Hello, I will talk to you all today about visualizing alignments using a tool called Integrative Genomics Viewer. Integrative Genomics Viewer is usually called IGV and can be used to visualize many different types of data in genomics. First of all, we will look at aligning long read sequencing data using a tool called Minimap2. Minimap2 is a fast aligner of long read sequencing data. We will learn how to interpret the output of Minimap2. We will load the alignment output into the IGV tool and visualize all of these alignments, and then we will look at how to identify small variants such as single nucleotide polymorphisms or SNPs and insertions and deletions, also called INDELs, in IGV. We will also look at the variant calling format files and also learn how to visualize these VCF files in IGV. Minimap2 is a specialized tool which is used to align long read sequencing data to a reference genome. It can also be used to perform whole genome alignment and for pairwise read alignment. The advantages of using Minimap2 over the algorithms that we have learned in class is that Minimap2 gives both fast and accurate alignments. It supports both the FASTA and FASTQ data, and you can align both DNA and RNA sequences using this data to a reference genome. Of course, you will have heard about a tool called BLAST. BLAST is a heuristic based alignment algorithm which uses statistics of occurrences of small k-mers in a database and aligns reads to a database and produces outputs. However, if specific k-mers occur very rarely in the database, then such alignments might be missed. Minimap2 only looks at the reference genome and the query sequence which needs to be aligned. The alignment that is generated using Minimap2 requires two inputs: the target sequence and the query sequence. In this case, the target sequence is the reference file and the query sequence is here, reads.fq, which is short for FASTQ. This output is passed on to a file, output.sam. We have two options over here, -a and x. -a tells Minimap2 to output a SAM file. We will learn what SAM files are. And -x map ONT optimizes several parameters of Minimap2 for aligning ONT reads. ONT reads are Oxford Nanopore Technology reads. These are third generation sequencing data from a company called Oxford Nanopore Technologies. ONT reads have specific characteristics which need to be taken into account by any aligner before aligning them to a reference genome. So we will perform these four operations on some sequencing data. Let me start the alignment first and then explain the command. We will be aligning the data in this file, mhcreads.fasta. I will pass the same argument as I have in the command, -ax map ONT. I will pass the CHM13 reference genome as the reference, and I will pass mhcreads.fastq as the query sequences. And we will pass this output onto alignments.sam. This will take some time to run, so I will go over the other commands first and then run the rest of them. We already know what minimap2 does. All of these other commands are intended to convert the output.sam file into a format which can be used by IGV. SAM Tools is a software which provides many different features for operations on SAM files. This command, SAM Tools view, converts the SAM file into a binary alignment mapping format. That is what is provided by this option -b S. We then sort the BAM file into a sorted form. This makes it easy for visualization tools to access data randomly, and we then also index it, which is also required by the visualization tool. We can see that minimap2 has started computing the alignments. Once these alignments are generated, we will convert the alignments.sam file into a binary alignment mapping format, and then we will move on to sorting it and then generating an index file for it. As this runs, let me give a brief explanation of what the sequence alignment mapping format is. A sequence alignment mapping format contains a few header lines followed by the alignments. Each of the alignments have 11 columns which are required, and any number of optional columns which may appear after these 11 columns. All of these lines are tab delimited, and each line will contain the query template name, a flag to indicate the different characteristics of that read, the reference sequence name, and the position of the leftmost mapping of the read. It is followed by the mapping quality and something called a cigar string. A cigar string gives information to the reader about how many matches followed by how many mismatches or insertions or deletions have appeared in the alignment. We will be using the SAM file and converting it into a BAM file before visualizing it using IGV. We can see that the alignment is complete. It took 142 seconds to run, and we can look for the alignment file now, and we can see that it is present. So we will run SAM Tools on it now. I have a separate Conda environment called SAM Tools to run the SAM Tools program. So I will use SAM Tools view on the alignments.sam file and pipe it over to a BAM file. Let's look for the BAM file. We will then sort this BAM file . Once we have sorted it, we will index it. We have two files now, alignments.sorted.bam and alignments.sorted.bam.bai. Both these files will be required by IGV for the visualization of the alignments. So let us see what the integrative genomics viewer is. Below is a screenshot. You can see here a screenshot of the IGV window, and you can see some alignments that have been visualized. So let's actually open up IGV on our own and visualize these alignments. So this is the IGV window, and you can see here this is pretty bare bones right now. You can see one track here which seems to have the genome labeled on it, and here we can see options for different reference genomes. Several hosted genomes are available which can be downloaded from the internet, and we will use the T2T CHM13V2 genome. Here you can select any particular chromosome. Let's look at chromosome 6, and you can see here a cytoband view of chromosome 6. Below here you can see different gene annotations, and when we load alignments we will be able to visualize them in this area. So let's go ahead and load the alignments. So click on File, Load from File, and I have already copied over these alignments from here. We have this in a file called demo.zip, and I have copied them over here. I will load aln.mhc.bam, and IGV will check for the existence of the index file. Okay, so something's appeared. We can see here the alignment.mhc.bam file in this corner, and it says zoom in to see the alignments. The reason for this is that the alignments are too small, the reads themselves are too small compared to the reference genome to visualize anything. So this is actually 170 million characters long that have to be represented in this region. We have specifically chosen to align reads from the MHC region of chromosome 6, and the MHC region of the human genome contains genes which code for immune response, and it's a very polymorphic region. That is to say, between any two humans we will see a lot of differences in this particular region of the genome, and this is a very important region clinically. So this region is specifically located somewhere between 28 million and 33 million. So that is to say somewhere in this region. Now whatever we have loaded onto IGV is called a track. All of these are genes which come preloaded. We will reduce this so that we can visualize our alignments better. And we will select, we have an option to zoom over here directly. We can also zoom by selecting a particular region on the cytoband. And while it does take a while, we can also provide specific coordinates directly over here. So let's say 28 million to 33 million. And you can see a 5 million length region available here. We still need to zoom in more to see the alignments. This is because alignments only become visible when the range of the genome being visualized is about 30,000. You can choose to edit that, but we want that this may slow down the response time from IGV. So let's zoom in, right? Let's look at, let's say, this region. You can even zoom into particular regions by left clicking a particular location here and then dragging over to the interval you want to visualize. Let's zoom in a little more. Right, you can already see some alignments that have been visualized, but let's look at 50,000 range of the chromosome. So you can see here that different reads are aligned to this region of the genome. We can zoom in all the way down and let's see what happens when we zoom a little bit further. Everything loaded by IGV onto its workspace is called a track. This is the coverage track and it gives information about how many reads are aligned to this particular position. You can see over here a chromosome and an index. Whenever you move your mouse over here, you can see that this number is changing and it actually indicates the position of the chromosome at that particular point. And the coverage track tells you at any particular point how many reads are aligned. So if we zoom even further, we are down to a 290 base pair range and we are actually seeing something interesting here. A sequence track has appeared here and this is actually what is present in the reference genome. If we zoom a little bit further, we now have a 41 base pair range and you can see here characters from the reference genome. Now you can see that everything is rather grey and this is because IGV does not like to show you information which is redundant. If something is grey in the alignment track over here, then what that means is that all the bases in this location are exactly the same as the reference genome. So whenever you see color, that means that something interesting has happened at that particular location. So let's zoom out a little bit and look for some color. To zoom out, you can click on this minus button here, or you can hold Alt and click in the alignment track. Now you can see a little bit of color over here, some green letters. So let's zoom in into just that region. Right. So you can see here, to move the alignment tracks around, just click anywhere on the track and then drag it to the left or to the right. You can see here that some reads have an adenine molecule represented over here. Some reads have an adenine molecule represented over here and some reads are still grey. You can also see that the coverage track is colored blue and green. If you click on the coverage track at this particular point, you will see a pop-up which shows the total count of reads that are aligned to that location and what nucleotides are present in those reads. So you can see 16 of them are adenine and 18 of them are cytosine. You can also see information about the forward oriented strands marked with plus and the reverse oriented strands marked with minus, and the counts of how many adenines and cytosines are present at that location. So let's see what the forward strands and the reverse strands are. If you right click anywhere on the track, you can choose to group the alignments, you can choose to sort the alignments, color them, or shade them. Let's enable shade alignments by mapping quality high. Clicking anywhere on a particular read, for example here left clicking, opens this window which gives information about the length of the read, the name assigned to the read, and the mapping quality and the reference span. The location is exactly the place where you have left clicked. So for example, if I left click this A, you will see that the location says at this 29,055,587, and you see that the base here is A and its quality value is 90. If you the bases themselves may be shaded lightly or darkly. The darker the shade, the higher the mapping quality. So let's right click here and sort the alignments by base first. What you see is that all the reads which contain A at this location have been sorted to the top, and all the reads that contain C, which is not displayed, have been sorted to the bottom. If you want to see what is here, then you can right click and select show all bases. This will actually enable all the bases everywhere, and this can be too much information. So we can just infer all the bases simply by recognizing what is in the sequence track. Now the sequence track is oriented in the forward direction. To see what is present in the reverse or direction, you can just click here, and this string gets reverse complemented. We will retain it in the forward direction. So let us look at sorting the alignments by read strand. You will see that some of the reads which contain A have shifted to the bottom, and let us actually color the alignments by read strand. And you will see that some have been colored red and some have been colored blue. The reads which have been colored red are in the forward direction, are oriented in the forward direction. The reads colored blue are oriented in the reverse direction. You will see in this last read here a 3. If you click on it, it says insertion of 3 bases. Whenever there is an insertion, it is indicated with an I or with a small number which represents how many characters have been inserted at that location. Any deletion will be represented with a gap in this form with a number indicating how many bases have been deleted. Now we know what single nucleotide polymorphisms and insertions and deletions look like when you visualize them using IGV. Let us look at what heterozygous single nucleotide polymorphisms look like. I will navigate over to a specific location . And you can see here at this center, some reads have thymine and the other reads have cytosine. Once again, let us sort these alignments by the base that is present at that location. And you can see in the coverage track that there are 29 reads aligned here, 13 of them are cytosine and 16 of them are thymine. This is a roughly equal split which suggests that there is a heterozygous variant present at this location. How do we know that this heterozygous variant is correct? Because the alignments that are generated by Minimap2 are not 100% accurate, and they are not necessarily the best possible alignment that can be generated given the alignment scores. Well, one thing to always check for is what we call strand bias. So if you right click here and sort the alignments by read strand, you should see roughly an equal number of thymines in both the forward and the reverse strand. Among the reads which are oriented in the forward direction, about half of them should contain thymine and the other half should contain cytosine. Again, for the reverse oriented reads, half of them should contain thymine and the other half should contain cytosine. This seems to be true in this scenario, and we probably have a heterozygous SNP at this location. When I say heterozygous SNP, what I mean is that of the 2 copies of the genome that are present in this individual, one from each parent, one of the parents has a thymine at this location and the other parent has a cytosine at this location. Let us look at another location. We can see here there are several different types of insertions. Once again, let us sort the reads by read strand. You can see some of these insertions are of length 15 and some other insertions are of length 21. And at all of these locations, you can see that there is a roughly even split of the 15 length insertion and the 21 length insertion in the forward and reverse oriented strands. So this is a heterozygous alternate form of insertion. Let us look at another location. At this location, we have some reads which contain adenine and some reads which contain a guanine. Let us sort this by read strand. And we see that most of the adenines are actually present in the reverse strand. This seems a little bit strange. So you should suspect that this variant may not be a real variant and there are some misaligned reads at this location. Let us look at what an insertion looks like. In this range, you will see that at this particular location, there are some reads which have a 7-character insertion and some reads which do not. Once again, we sort the alignments by read strand and we see a roughly even split. Similarly, you can visualize deletions as well. Now that we have visualized alignments using IGV, let us see what variants are. We know what single nucleotide polymorphisms are. They will appear as single base edits in the alignment track. Remember to always check for strand bias and also check the alignment quality by selecting the reads. When you look at insertions and deletions, they will either be marked with an I or a hyphen on the read respectively. Again, remember to check for strand bias and read alignment quality. Now, there are specialized tools which can identify variants based on alignments to a reference genome. One such tool is a deep learning model called DeepVariant, and it is used for accurate small variant calling. DeepVariant is usually run from a Docker image and it outputs a VCF file. I will not be executing this program here, but I can show you the output file which is present here. Once again, to visualize the variant calling file, you will need to index it, and you can use a program called tabix to do this. Let us load the VCF file onto the IGV for this set of reads. I will select this gzipped VCF file and load it here. You can see that another track has appeared here called variants.vcf.gz. Let us zoom out quite a bit to see what this track looks like at a low resolution. Before we look at this file, let us look at what a variant calling format file is. A VCF file has meta information at the top, which always start with 2 hashes, and they contain information about all the other data that is present in the VCF file. They have a header file which describes all the different columns which are present in this VCF file, and this header file will contain the chromosome, the position, the ID, the reference allele, the alternate allele, and the quality of the variant call, and whether it has passed a filter or not. Some additional information is stored here, and for example, AF stands for the allele frequency, which is how many, the fraction of the alternate allele as compared to all the reads which are aligned to that location. The format is actually used when you have other when you have other samples, and you can use this to describe a lot of information. For example, GT describes the genotype of that particular variant, and what pipe 0 says is that the variant in NA0001 is the reference allele in both haplotypes. NA0002 says 1 pipe 0, which means the alternate allele in the first haplotype and the reference allele in the second haplotype, and so on. So let us look at the VCF file. So you can see a lot of marks over here. Each of these marks correspond to a variant that has been called. Some of these are dark, which means that the variant has, selecting it says, gives this dialog box which tells you some information about the variant at that location. And some of these are lightly shaded, and you can see that that's because they have been filtered out. Now let's look at one such example. Let us zoom in on this region of the genome. When zooming out, you will notice that the alignments have moved apart, and we can just repack the alignments and get them close once again. We can just sort the alignments by start location, and they come, and you can see that they are all close together now. Let's zoom in on this variant here. You can see here we have 21 reads aligning to this location. 8 of them have an A and 13 of them have a C. And if you sort these alignments by read strand, you will see that we probably suspect a heterozygous variant. Let's see what DeepVariant says. To see what DeepVariant says, you can just click on that variant in the variant track, and you can see that at this particular position the reference is a cytosine and the alternate allele is an adenine. The quality of the variant call is 7.6 and it says that it's a SNP. Let us also look at an insertion. So at this location you see a small mark over here which corresponds to the insertion. This is the same insertion that we had visualized earlier, and you can see that the reference is A and the alternate is either this or this. These two, and what has happened here is that both of the alleles that have appeared in the sample are alternate alleles. The quality of this call is 32.5 and it has not been filtered out. Okay. So we have learnt how to align reads to a reference genome using Minimap2. We have learnt how to generate a SAM file and convert it into a BAM file using SAM tools. We have loaded the BAM file onto IGV, visualized the alignments and identified single nucleotide polymorphisms and insertions and deletions by eyeballing the alignments ourselves. We can confirm our guesses by using a variant caller such as DeepVariant and load the VCF file that has been generated by DeepVariant onto IGV. We have learnt the different features of the IGV tool for visualizing alignments and variant calls, and that concludes this demonstration. Thank you.